This conference will now be recorded.
All right, folks, so good morning. Thank you for attending today. We are going to be looking at and reviewing information regarding Chapter 31. And so let's look at this first slide here as we start off our class today. So why does it matter that we study this information here as far as in Chapter 31 regarding phylogeny for animals, coelomates, and protostomes? So zoologists have described nearly 2 million living species in the kingdom Animalia. Right? So we're going to be looking at and discussing the terms vertebrates, as far as invertebrates are those without a spine, without a backbone. Vertebrates have a backbone. About 67,000 of those 2 million would be considered vertebrates. Okay, important statement here as far as the evolutionary diversification of animals resulted from their ability to consume other organisms as food, and for most groups their ability to move from one place to another, right? So their ability to not be just, now there are some animals that in later stages of development are immobile, they're not able to move. So know that there, so that's important, and that can also affect their ability to access food and such. But we're going to see in a moment here as far as the term heterotroph, right? So the term heterotroph means that they are not producing their own food, right? So we looked at plants and discussed plants and looked at as far as photosynthesis is concerned, and so they're autotrophs. So heterotrophs, right? The animals eat other organisms to acquire energy and nutrients and such. And so very important to have that distinction as far as what is an animal. Eukaryotic organisms. So we have cells that have membranes, membrane-bound organelles, a nucleus, right, containing the genetic material for the cell. Multicellular organisms, right? So we're not talking about a unicellular organism. We're talking about multiple cells comprising this animal structure. You'll see here, unlike plants and fungi, cells lack cell walls. So that's another difference there. So we have a plasma membrane that encompasses the contents of the cell comprised of the cytoplasm and the organelles and such in the nucleus. What else we're seeing here in large organisms? Okay, yeah, so specialized tissues and organ systems deliver nutrients and oxygen to cells and removal of waste, right? This is a part of what the animal can do and as far as its functions. And we'll see as far as looking at a little bit more detail regarding the tissues and such. So actually, no, and this was from what we had studied before there as far as, let's see. When we start off the beginning of the semester looking at human anatomy and such. Structural. And so if you recall, let's see one that we have here. Okay, I guess we can look at this here. Sure, this is good. This is as good enough image as we can see here as far as the structural organization regarding the human body, the levels of organization. So you see here from the chemical level, right, we get here to the cellular level. So let's go back over here. We'll start here first, because then we'll come around and see that we have the human organism, right? So here's the organism. It's a little cute little child, right? This child is comprised of, her body is comprised of multiple organ systems, right? And so you'll see here the digestive system is just an example of. So this organism is comprised of organ systems. These organ systems are comprised of organs. The organs are comprised of tissues. These tissues are made up of and comprised of cells. And these cells are made up of, right, chemicals and such, and all these DNA and these subatomic particles and such. So we recall this, right? And so today, as we're looking at and discussing what it is to be an animal and such, and what that represents, we realize that, again, cells and tissues, right, and organs and organ systems and such, comprising these animals. And again, there's going to be from very simple animals to more complex animals and such. And we're going to see that also in this PowerPoint today. You'll see here that animals use oxygen to metabolize food. And we would say the metabolic processes that take place, aerobic respiration, so the need for oxygen in order to produce ATP and produce other energy compounds and such that are necessary for cell life and such, and also produce processing of nutrients and such. All animals are motile, right, at some time in their lives. Now, this is important to see that, so because we're going to see some organisms that are animals that are sessile, they're immobile, but that's in their adult stage. So in some species, the young are motile, right? So they have movement, and adults are sessile, so they are immobile. So that needs to be kept in mind also regarding the diversity present within the kingdom Animalia. Again, 2 million living species present within, invertebrates, vertebrates. So having that understanding there, you'll see here as far as the ability to reproduce, whether asexually or sexually. Now, you're going to realize and think that many of these animals present within the animal kingdom will sexually reproduce, right? And we've talked about, right, ad nauseum regarding when we were looking at the human body as well in the beginning of the semester, as well as looking at the plants as far as that there are these gametes, these sex cells, the egg and the sperm. They are haploid as far as in their presentation of and their contents of the genetic material present within. Once they come together at fertilization, again, they form this diploid zygote, okay? A complete cell with a full complement of 2 pairs of the chromosomes that are needed for the organism. Zygote develops into a multicellular embryo. This embryo then becomes an immature juvenile or a larva, right? Which develops into a reproductively mature adult. Okay, so simple enough. And here you're seeing, and these are different types of, and I'm going to reduce this for a moment so you can see. So let's go here. Animals of the Burgess shale. So Apobinia had 5 eyes and a grasping organ on its head. It almost looks like something out of a Doctor Who or science fiction type of a program. Hallucigenia, quite an interesting name, had 7 pairs of spines and soft protuberances. Right? And so I do have some fossils of different organisms that you look at them and you go, I can't even believe that they would be around and alive. And yet we have in the fossil record these representations of these organisms that were alive many years ago, and pretty cool to see them. And again, it's one of my most fun hobbies is to collect fossils and go to museums and look at fossils and such. And I just, as much as I love biology, the study of living organisms, I do love also the study of fossilized organisms too. It's just fascinating. So you'll see here as far as animal origin, so common ancestry, right? So common ancestor of all animals was probably, right? So again, these are just hypotheses and just thoughts regarding what they would, as far as evolutionary theory is concerned. So we're looking at a Coano flagellate, probably lived approximately, and again, according to evolutionary theory, 700 million years ago. Ernest Haeckel proposed that, so you'll see here as far as now these 3 propositions here regarding the common ancestor. The colonial flagellated ancestor was a hollow, ball-shaped organism with unspecialized cells. 2, the cells became specialized for particular functions. 3, the specialization then and as far as related to these functions then also developed these reorganization, producing layers, okay? A sac within a sac type body plan. And we're going to see this as far as what's going on as far as the layers, the germ layers, as far as animal structure is concerned and such. So here you're seeing as far as this shows you, so many biologists agree that animals arose from a colonial flagellated protist in which cells became specialized for specific functions, and developmental reorganization produced these layers, right? These layers. The cell movements illustrated here are similar to those that occur during the early development of many animals. All right, and so you're seeing here, and we're going to see in a few slides forward at the end of this presentation today, these germ layers and such. And so you're seeing here that these, here we have all these cells present, multicellular, and we have also cells that are then going to be become specialized with certain functions associated with that specialization. And then there's going to also be development, so this developmental reorganization, so that there's going to be these different spaces present within this organism and such. Okay. So key innovations in animal evolution. So several key morphological innovations, right? So these presentations that are going to help, and as far as we're used to develop hypotheses about the evolutionary relationships of the major animal groups. So tissue structure, symmetry, cavities, developmental patterns, and segmentation. And so that's what we're going to take our focus on today as far as for these next 33 slides here forward, okay? So as far as tissues are concerned, so we already I shared that image with you just before there regarding as far as human structural organization is concerned, and seeing that these tissues are comprised of cells with a common structure and function, right? And so, if you recall, when we were looking at human anatomy, we talked about epithelial tissue, we talked about muscular tissue, we talked about connective tissue, right? So these different types of tissues are comprised of special types of cells that allow for this tissue to have its certain functions and the structure present in order to provide some type of, whether it's an organ present with an organ system, whatever it may be, whether it's a tube. Think about this as far as human structure is concerned. We're comprised of these tubes present within the body. So, the different organs present within, they're just expanded tubes, are they not? With special types of cells and special types of secretions that are produced in order to protect or provide some type of function, right? So quite fascinating. Now you're going to see here two terms that you'll need to know as far as parazoa and eumetazoa. Okay? So when we look at the eumetazoa, we're looking at, they have tissues, and these are all the other animals other than sponges. Okay, so when we look at the kingdom Animalia, so sponges lack tissue. All the other animals have tissues. Okay, so in the eumetazoan development, embryonic tissues form either 2 or 3 primary cell layers. Now we're going to see that most are triploblastic, right? So having 3 primary cell layers, and that's what we're going to be looking at in the next slide here. And when you think of human development, there's going to be this ecto, meso, and endoderms, but we're going to see this in most of the animals present within the eumetazoa. You're going to have these 3 germ layers. And these three germ layers will differentiate and become the different structures that comprise the organism. So you'll see here, as far as the germ layers, these primary cell layers are concerned, so the ecto, meso, and endoderm. So you'll need to know that the ecto, outermost layer, the meso, it's the middle layer, the endo, the innermost layer. And now what occurs, what actually develops from these layers, right? So you'll see here as far as the ectoderm, the external covering, and the nervous system. So the neuroectoderm develops from that outermost layer. The mesoderm, so you're thinking as far as the muscles. So you see how the M's kind of correlate and go together? Mesoderm, middle layer, muscles of the body, and most of the other structures between the gut and the external covering. And then you have the endoderm. So the endoderm is the inner lining, the innermost layer, develops into the lining of the gut, right? The digestive system, in some animals, respiratory organs also, endoderm, okay? Provided some more additional information as far as ecto, neurecto, right? So this these next 2 slides I took from, actually next 3 slides I took from another Cengage product that I teach as far as human biology. And so you'll see here as far as the ectoderm, neuroectoderm, this is all the outer layer of the germ layer. So the mesoderm, that middle layer, muscles, bones. You'll see here endoderm, organs, glands. This is for human development as far as the germ layers are concerned. Okay. And here's just a representation too, just simple image here to see as far as ectoderm, outer layer, mesoderm, middle layer, endoderm, this inner layer here, okay? These different germ layers present. Here's just another image showing you the ecto, meso, and endoderm in this image here, right? A developing embryo. And this I like as far as this was just pulled off of the web here. You'll see their reference to who was Stem Cell Thailand as far as just providing, but this is in other texts and such also, but showing you ecto, meso, and endoderm as far as what's being produced as a result of this germ layer, So you'll see here as far as the different structures and such, and so don't have to memorize this or know this, but just know the basics regarding ecto, meso, and endoderm. I just provided this for additional information for you all. Again, this is for human anatomy. Now, body symmetry. So when you look at another human, right, and realize that there many have symmetry, but there are some that do not. And right away, you recognize that. And as far as so having spent many years as a chiropractor taking care of patients, I've had patients in my office that are missing parts of their body, and as a result of catastrophic injuries to cancer, other disease processes and such, or as a result of actually developmental issues in the womb, right? So congenital issues they're born with, they're missing a limb, fingers, so on. We recognize this, it's important, and know that the thyroid hormone. So when we're thinking of hormones are the chemicals that really have specific purposes within the body to maintain different types of metabolic activities present within the body that help to maintain homeostasis, a stable environment within the body, no matter what goes on outside of the body. Thyroid hormone, really, as far as developmentally is concerned, it works in conjunction with growth hormone in order to produce symmetrical limbs and such, and symmetry throughout the body. Quite interesting. And so we do see, when we do recognize, so asymmetry as far as, let me show you an image, and I believe that I might have showed this to you all when we were in the beginning of the semester. It's called Proteus syndrome. This is just one in particular type of genetic issue where patients will have issues where, so it's not easy to look at, folks, and I'm not trying to, give you an issue, but these are real people who have developmental issues where you'll see here as far as that, this is a hand, folks, and it's quite sad indeed to see that folks suffer with this asymmetry that can go on and take place as developmental as development takes place within the human body and such. This is just one aspect called Proteus syndrome. This woman here, who has since had both of her lower extremities amputated and such, look at how the upper torso, right, from the waist superiorward, is really developmentally looks fine, and yet her lower extremities were growing out of control and asymmetrically. So symmetry is very important, okay? So as we come back here to this slide, so sponges. So again, when we're looking at the, back here, so parazoa in comparison to the eumetazoa, parazoa for the sponges, eumetazoa for all the other animals. Irregular shapes, asymmetrical, okay? And I've never had the opportunity to go scuba diving or snorkeling and such in a tropical area, but to be able to watch, video of folks being able to do this and see the different types of sponges that are cool, colors and presentations present, quite remarkable. Now, know that most animals are symmetrical, right? So their bodies can be divided by a plane into mirror image halves. So we can take the human body and do a mid-sagittal cut through the human body and create a right half and a left half, right? And they're mirror images of. And know that if folks are as a result of injury, as a result of whether that be through some type of like a soldier, right? Dealing with an injury, or someone who was involved in a workplace injury, or primarily those. Yes, yeah, indeed. That's sorry. I'm just thinking about it for a moment there. Yeah, those type of patients can have issues with phantom pain. Phantom pain deals with you're missing a portion of your body, and yet you still experience pain and discomfort as a result of that image, that part of your body. Your brain is still functioning as if it's still there, and giving you and causing you discomfort, even though it's not there. And so they'll do mirror image type therapy in order for you to actually so mirror therapy. So let's look at an image here. And so this is quite interesting in that. So here's a gentleman who does not have a right upper extremity as far as their hand. And so what they're looking at is they're looking at this mirror, and the mirror image of their left present arm to help them to be able to focus on what's missing as far as their right side is concerned. Mirror box therapy, mirror therapy. And so this really helps as far as rehabilitation, and as far as those that are missing limbs and such, that they can help the brain to and I don't recall, I'm sorry, the physiology present in how this actually works, but it does over extended period of time and through therapy to be able to have less pain and discomfort, like I said, as far as that phantom limb pain is concerned. So symmetry is very important. So again, most animals are symmetrical. The humanozoans, right? We're talking about all animals other than sponges exhibit one of two body symmetry patterns. And so when we think of bilateral, right, we're thinking of bilateral symmetry. So we have a right and a left side, and they're equal. We have a right and a left upper extremity, lower extremity, right? Our face as far as the right side and left side. Now, a patient can suffer from some type of stroke that could damage one side of the musculature that's not having control over, or they can have also something called Bell's palsy, where we believe a virus can be the etiology of that, and it can cause symptoms very similar to a patient having a stroke, but not affecting the whole body. So just affecting just the face as far as the facial musculature and those muscles that control the face. So getting back to here, so bilateral symmetry, left and right sides are essentially mirror images of each other. Radiata, or radial symmetry, deals with body parts that are arranged around a central axis, like spokes on a wheel, okay? So that's as far as the presentation, as far as symmetry is concerned. So bilateral symmetry. Now this is also important to see as far as we're going to talk about terms in order to describe areas of the body and such, and of animal organisms. You'll see here cephalization. So cephalization, so the development of an anterior head where sensory organs and nervous system tissue are concentrated. And when you look at a human, right, do we not see that we have our senses for vision, for smell, right, olfaction, for our ability to eat through our mouth and the sensation of taste, and also for sense of hearing and such. These are all anterior focused. And so what do I mean by anterior? So bilaterally symmetrical animals have an anterior. So when we think of anterior, we think of the front of the body. Posterior, we think of the back of the body, right? We can also use terms like dorsal and ventral. Dorsal and ventral also can mean dorsal, can mean posterior. Ventral can mean anterior. We can also use those terms as far as when you think of dorsal, where is the dorsal fin on a shark? It's on the back of the shark, right? So we can use those terms for that. So important to know those terms, okay? So here we're looking at, as far as dorsal, as far as ventral is concerned, anterior and posterior. So here we have that radial symmetry. So here's that bilateral symmetry, radial symmetry, okay? Very important to see that there, and pretty cool. So most animals have either radial or bilateral symmetry. So the terms of coelomate and pseudocoelomate and coelomate, right? So you'll have, these are regarding cavities of the body. So acoelomate animals have no body cavities, right? That separates the gut from the muscles of the body wall, okay? Pseudocoelomate, so we're thinking of this pseudocoelomate, fluid-filled space between the gut and muscles of the body wall, okay? So no cavity, fluid-filled cavity. Coelomate, so body cavities, so the coelom, right? Completely lined by membranes, right? So we, and as far as thinking of the human organism, right? We're coelomate. We have these body cavities. So we have these cavities that are lined. So I have we have a relative who is experiencing peritoneal dialysis. And so what goes on with that is that in the peritoneum, in the coverings of the abdominal pelvic region, right, in the abdominal pelvic cavity, there's going to be a fluid that's going to be put within the peritoneum in order to exchange with the blood, actually removal of toxins, of waste. Their kidneys are not functioning properly, his kidneys are not functioning properly, and as a result, he needs to go through this dialysis in order to remove the waste products from the blood, in order to then, at the end of the night, when he wakes up in the morning, be able to have this exchange, actually this fluid, leave his body. I'll show you this, it's quite interesting. So here we have this peritoneal dialysis, and what's going on is that, let's open this up here. So we have here that, and let's make this bigger. So at night, when he's sleeping, he has this dialysate enter into his abdominal pelvic, the peritoneum, into this cavity, this peritoneal cavity. You have organs present here, right? So you have your abdominal and pelvic organs present here. They're covered. Now, the kidneys are retroperitoneal. They're outside of this peritoneal cavity. But you have these organs, and there's a lot of vasculature present. And what's present within this solution will interact with, not enter into, okay, so know this, it just enters into the space here surrounding all of these organs and the blood vessels that are present. And because of what's present within, as far as solutes are concerned, within this solution, it's able to pull out from the blood vessels the waste products. So waste fluid present within this dialysate over an extended period of time. So he's sleeping at night, and then when he wakes up in the morning, they'll actually drain this fluid from the peritoneal cavity and enter into this bag here where it'll be disposed of, okay? And the waste products from the blood will be removed. Is this as good as your kidneys being able to filter the blood in order to remove waste products? No, it's not as good. But this is a method that we can use in order to keep someone alive, because without properly functioning kidneys, you'll die, okay? So, and folks, and this kind of a thing, as far as you know, issues with the kidneys functioning and lack of function, this can affect other animals also, not just humans, but we have the ability to be able to do this type of peritoneal dialysis in order to remove the waste products. And again, so the peritoneum, this is just the cavity. So let me show you another image here, as just far as the, and so let's go right here. So here you're seeing, folks, as far as this image, and let me just make it bigger. So it's just showing you a mid-sagittal image, right? And here you're seeing as far as that present within the peritoneal cavity, these organs are present. They're just showing you a cross section, right? And then you'll have, as far as present within this peritoneum, the organs will be present and they'll be covered, okay? And it just keeps the areas of the body to itself. Here's another image also. Let's see, this is maybe a little bit easier for you to see. Yeah, here we go. And so here you're looking at and seeing that we have different organs present within the abdominal pelvic region that are covered in this peritoneum, this actual serous membrane covering. And that's what you're seeing here as far as where it says here membranes, right? So these membranes actually are covering the structures present within the body, okay? So body cavity is coelom. So coelomate animals have body cavities, they have the coelom, completely lined by membranes, right? Derived from the mesoderm, that middle layer, that middle germ layer, okay? Extensions of the peritoneum, the mesenteries, surround the internal organs and suspend them within the coelom. And so those mesentery are present and they are highly vascular, okay? And this is how we can do this peritoneal dialysis and such, because there's a lot of vascularity and blood vessels all surrounding the tissues present within the peritoneal cavity. So body cavities. So body plans of the pseudocoelomate and coelomate animals have formed tube within a tube. And so realize that as the early developmental stages, there are tubes that develop, and these rudimentary tubes get more complex as the development takes place. So we have, let's show you here. So our little granddaughter that's not born yet is 20 weeks old. So she's 20 weeks old. So where are we at as far as, so that's, so 20-week old, you can see how tiny. Pretty wild stuff, folks, as far as the layers and such, and as far as the development that occurs at 20 weeks. So think about 20 weeks. So we're 5 months approximate, right? So we're talking about 40 weeks gestation so halfway through the pregnancy and such, our little granddaughter, and pretty remarkable how even at 20 weeks, what's going on as far as development is concerned, and look at as far as looking at the structures and the symmetry that's present, and the continued development will take place as far as the complexity present within, as far as the organ structures and such. Pretty wild stuff, folks. So let's look here, get back to here. So this tube within a tube, these body cavities, the digestive system forms the inner tube, the body wall, outer coverings, the outer tube, fluid-filled body cavity lies between these tubes. And again, the complexity of the tubes and their structures, just as development goes on until birth occurs, just remarkable. Incompressible fluid within the body serves as a hydrostatic skeleton, which provides support. So not only just the bony skeleton, right, that you would think of when you think of skeleton. So take a look at here for a moment. I provided this slide for you all to see. As far as the skeleton is a framework that shapes and supports an animal, right? Protects its internal organs, allows for it to move in its environment. So we're looking at as far as the endoskeleton. So this is what humans, we have, the skeletons present within, not on the outer part. Exoskeleton, think the shell outside a snail, insects and such, exoskeleton. Hydrostatic skeleton, this present within, so are there bony structures present within this this earthworm, this segmented worm, right? No, not at all, right? So there's water that's present within under pressure that will allow for it and to provide support and protection of the structures present within. So endoskeleton, right, consists of bones, cartilage, muscle, tendon, ligaments, right? Exoskeleton, outside, so heart, outer shell. Hydrostatic skeleton, fluid-filled cavities surrounded by muscle, right? So this is as far as thinking of a worm is concerned, the hydrostatic skeleton. as far as cavities are concerned. So a coelomate, coelomate and pseudocoelomate, right? So here's that segmented worm, right? The coelomate, right? Humans, coelomate. We have these multiple cavities present and covered in serous membranes, these membranes that cover the structures present within. You'll see here as far as the pseudocoelomates and the acoelomates, these can be like parasitic type organisms, whether they be very tiny and microscopic, or you can actually see with the visible eye, but different as far as the different cavities present within. And here you're seeing as far as a breakdown and showing you as far as acoelomate animals, no body cavities, separates the gut and the body wall. Okay, you'll see here as far as and that's a flatworm represented. You'll see here as far as the pseudocoelomates, right? So you'll see here as far as forms between the gut and the body wall, pseudocoelom, right? And so here you're seeing that represented here in the green. Okay. And then as far as coelomates are concerned, and realizing that we have, so here's the spine. So here's the posterior, the dorsum, right? Here would be the anterior, okay? And you're looking at here, this would be more of a horizontal cut through the human, and looking at the different structures present within. So here we have as far as the cavity is concerned, so the peritoneal cavity and structures present within it. Now, these terms here as far as coming back to developmental patterns. So bilateral symmetrical animals are divided into two lineages. So this is the animals now grouping that are bilaterally symmetrical. We have protostomes and deuterostomes. Okay, deuterostomes. So protostomes, most phyla of invertebrates, right? No vertebra, no lack of spine. Vertebrates and their nearest invertebrate relatives would be considered deuterostomes or deuterostomes. Now, this term cleavage has to deal again with what's taking place as far as mitotic division, right? So meiotic division, meiosis, produces the sperm and the egg, okay? The gametes, the sex cells. Now, mitotic division, this is what takes place with all the other cells present within the body. And so when there's a fertilized egg, so this becomes now a complete cell, right? Complete cell, the zygote. And so mitosis takes place as far as mitotic division will help to differentiate then over time the different structures that are present within the organism. So there's spiral cleavage for protostomes and deuterostomes, radial cleavage. Okay, you'll see here as far as this is just giving you an example and showing you as far as the radial cleavage is concerned, as far as the spiral cleavage. Just know that term there and just realize that that's one of the examples of a difference between protostomes and deuterostomes or deuterostomes. As far as also protostomes and deuterostomes, what's developing first as far as the different aspect of the cleavage and the differentiation of the meiotic division of the cells and of the tissues and of the germ layers, right? So you'll see here as far as the mouth develops first and then the anus. As far as the deuterostomes, it's opposite. The anus develops and then the second opening is the mouth. So after cleavage, after these changes during mitotic division of the embryo here, some cells migrate inward through the blastophore, this opening, and become the endoderm, right? The endoderm, again, that inner portion of the structure of the body, that germ layer, which forms the developing gut, okay? And then the second opening at the opposite end of the embryo turns this developing gut into a digestive tube. And then you're seeing as far as what's the beginning and the end of the digestive tube, right? The mouth, the oral cavity, and the anus. And here you're just seeing as far as those are concerned, those regions. Okay, and here you're just seeing again two more changes taking place within the germ layers. These slides really, folks, not any questions coming from this. Okay, but here last slide, yeah, last slide as far as segmentation. So knowing that, right, So some proteostomes and deuterostomes exhibit segmentation. Okay? The production of body parts is repeating units. Now, in humans, other vertebrates, right, seen as far as in the mesodermal tissue. So the vertebral column, the ribs, the associated muscles, segmentation. That's an example of present within humans. As far as in other animals, such as earthworms, segmentation also is seen in structures derived from the endo and ectoderm. Okay? So folks,